But I'm going to start by introducing the very fundamentals of the digestive system and, again, what we need to know for today's lab. And then we'll expand on all of these ideas on Wednesday. So basically, the digestive system, you are eating stuff, the stuff that you're putting into your mouth in general is going to be the bodies of other organisms. It could be plants. Maybe it's some broccoli, or maybe it's a wheat plant. Those are organisms. Or maybe it's like, some bacon. A little piggy. So you're basically taking these other organisms that have assembled all their biomolecules in whatever form they need to make a pig or a broccoli plant or whatever. And there's going to be two steps for your digestive system. One is you're going to have to, break down food, food. This is what we call food, these things that these other organisms that you're eating. Break down food, basically all the way down into the building block molecules. So that's the breakdown. This thing that used to be a pig or used to be a broccoli plant is going to get dismantled into individual monosaccharides and amino acids and fatty acids and things like that. And then part two, you've got to absorb those into your body. We are not going to look at the absorption right now. We're going to be focusing on the breakdown for this lab. Right, and it's a really trippy thing if you think about what is digestive system. What do animals do? Plants actually make some sugars from sun, but all the heterotrophs, they basically consume other organisms, dismantle their bodies, and then repattern those molecules into their own body. And then something else comes and eats that. It's like, we eat the pig and dismantle the pig molecules and to use our DNA code to reassemble them into a human body. And then, if some tiger eats you, or even if you just die and lie there in the bacteria just dismantle your flesh into building block molecules and repattern them into little bacterial cells. It's kind of trippy. Everything's just eating and consuming itself and repatterning those and getting consumed in subsequent, stages and getting repatterned by whatever consumed it. So let's back. There we go. So this is the digestive tract. The digestive tract starts with your mouth. You got the esophagus coming down. There's the stomach. There's the small intestine, the large intestine, finally out the anus. So this is the GI tract. It's the tube through you. Again, people say people are like a donut. It's got a hole at the top and a hole at the bottom and a tube, hole running through. This is where the breakdown occurs. When we get to the absorption is going to be, across the walls into the body. But that's the story for Wednesday. For today, we're going to look at the breakdown that's happening in this. We're going to look at 3 different regions that breakdown occurs and 3 different main kinds of biomolecules that are being broken down. We're going to start in the mouth. And we're going to also look in the stomach. We're also going to look in the small intestine. And in each of these regions, we're going to look at a particular kind of molecule. We're going to look at carbohydrates breaking down in the mouth. We're going to look at proteins breaking down in the stomach. And we're going to look at lipids breaking down in the small intestine. In reality, the small intestine also does everything else. So we are focusing on lipids, but when we talk about digestive processes in more detail on Wednesday, you'll see the small intestine does everything. So don't think of the small intestine only doing lipids. It actually does proteins, it does carbohydrates, it does nucleic acids. Okay. So how are you going to dismantle starches into simple sugars? How are you going to dismantle proteins into amino acids? What do we use to do things like that?
Enzyme.
Enzymes, exactly. So we're going to be looking at the enzymatic breakdown of these different nutrients and what enzymes in your body are doing that. So in your mouth, we have saliva. And saliva has what we call salivary amylase, which is amylase breaks down starch, enzyme to break down starch. Maybe let me put, actually I can write it here. So saliva has salivary amylase, which is this enzyme which breaks down starch. It also has mucus. The mucus is important to just bind like your food. Here I've got like some little pretzel fish, right? I don't know if you can see, it's like dust. If you chew up these pretzels, they're just like dust. You're not going to swallow them. You're going to choke if you try to swallow dust. So the mucus is important part of your saliva as well, to kind of bind the food into a slippery little bolus, little ball that you can swallow. So we actually are going to look at 2 aspects of saliva. We're going to see that it does have mucus. And then you'll also watch salivary amylase in action, which breaks down carbohydrates. And carbohydrates are going to break down into what?
Glucose.
Yeah, so ultimately glucose. Although in this lab, the first stages is going to be like smaller chains, so like maltose, which is like 2 glucoses together. So that's going to be part of our lab. In the stomach, we have a enzyme called pepsin, which is a protease, breaks down proteins. And in the small intestine, we're going to have a lipase called pancreatin. So in each case, we're going to have an enzyme. The enzyme is either salivary amylase, breaking down the starch, the carbohydrates, pepsin breaking down the proteins in the stomach, and pancreatin, the lipase, breaking down the fats. What do we know about appropriate conditions for an enzyme to function well? So what kind of molecule is an enzyme?
Protein.
It's a protein. And so we want to really make sure that the protein does not denature. Remember that denaturing, losing its shape. So we need to make sure that the protein is not denatured, because if it denatures, then it doesn't bind the substrate, it doesn't do its job. What kind of things can denature a protein?
Temperature.
Say it again, Ann.
Temperature.
So temperature.
pH.
We'll say it again.
pH.
pH. So pH is something we are going to play with in our lab. So what would be the normal pH in your mouth?
I'm going to guess it's the same as blood, like 7.4.
Yeah, it's pretty neutral. So this amylase is going to function well under kind of neutral conditions. You know, in our lab, we're going to have one of the tubes where we add concentrated hydrochloric acid, see how that affects how the enzyme can do its job. So you want to definitely think about how pH of the environment affects the ability of an enzyme to work. In the stomach, where it's super acidic, normally, this pepsin is actually optimized so it has just the perfect shape when the pH is really low. So this is going to actually denature when the pH is more like neutral, right? So, you know, and in this one will work fine in kind of more neutral conditions. But thinking about pH, particularly in when we're looking at the protein digestion and in the carbohydrate digestion, we are going to be playing with pH as a independent variable. And so you definitely want to think about how that's going to affect the final results of trying to digest using that enzyme. The other thing we need to add in here is in lipids. So lipase breaks down fats. What is different about lipids compared to carbohydrates and proteins in terms of just sloshing around in the watery environment inside your digestive tract?
They're insoluble.
They don't, yeah, they're hydrophobic. They don't dissolve in water. So if you have in fact, I'm going to go to a picture. I'm going to share a picture right now. You know, if I have a big fat glob in that I've eaten, let's say I just ate a big thing of butter or something. You know, if I have my enzyme is trying to attack it, it can come in just along the edges, but it can't really get to the majority of the fat because it's just, you know, protected inside the middle of the globule. So even if I have an enzyme that's trying to break down the fat, it can't get at most of the fat molecules. And if you shake things up, it'll just reclump, right? If you take oil and vinegar, oil and water, and you shake it, it comes apart, but then it regroups. So we need a way to make this more accessible to the lipase, to the enzyme that breaks down the fats. And that's where the bile salts come in. Bile salts are amphipathic, just like we talked about with like the phospholipids. They have a hydrophobic side and a hydrophilic side. So if you have your fat droplet, normally your fat droplets would all just regroup back into one big fat droplet. But instead, these bile salts, kind of like surfactant we were talking about, it kind of gets in where the hydrophobic side goes against the fat droplet, the hydrophilic side goes near the water. And so all of the little fat droplets just stay as dinky little droplets. And it's called emulsification, emulsified fat droplets. And now we have way more surface area for the enzyme to be attacking. I have like one, I can show you one more. I just like went online and like was trying to find these things. Right, so you initially have a fat glob and the bile salts, and then as the fat gets kind of broken apart as you're mixing things up, the bile salts go in between and make sure that the fat droplets don't regroup and stay as dinky little droplets. And then those dinky little droplets are much more vulnerable to breakdown by the lipase, the lipid breaking down enzyme. So that's going to be part of today's lab is, so let me, one quick, where are we? We're good. So we're going to basically be looking at carbohydrates. We're going to be looking at protein. We're going to be looking at lipids. And in each case, we are going to have kind of our representative food thing we're going to use for that. For carbs, we're just going to use a starch solution. We got starch. For protein, we're going to use egg white. For lipid, we're going to use cream. And just as a heads up, cream is already emulsified to some degree. Obviously, cream has the kind of fat spread out in the water. So we are going to be able to further emulsify it, but it's not like cream is pure fat. It's actually already an emulsion to some degree. So we have our food stuff. We're going to have our enzyme. Right here, we're going to have the amylase from your saliva. Here, we're going to have pepsin. Here, we're going to have pancreatin. We need some way to assess breakdown. And here we have different things in the three different cases. The simplest one is the protein. For the protein, you're going to cut the egg white into identical little squares, 7 millimeters per side. And then you can just visually inspect after like an hour. Is it smaller? If the egg white is digesting, it's going to be dissolving and the square is going to look smaller. So you can look at a square that hasn't been treated and then look at one that has and see if the square has gotten smaller. That is going to tell you that protein breakdown has been happening. It's one of these more qualitative assays, right? For the lipids, we're going to use what we call Litmus Cream. So litmus, do people know what litmus is?
No.
So it's a pH indicator. So litmus cream. So basically this is red when it's acidic, blue when it's neutral or basic. So when we just mix this litmus powder into the cream is pretty neutral, so it stays blue. What do fats break down into?
11 a.m.
Do you remember what a fat was? The fat, the triglyceride?
Three fatty acids and a glycerol?
Exactly. So it's 3 fatty acids, which means that as you break down the fats, we're freeing up fatty acids and the pH is going down. So that is going to be our test for the presence of fat breakdown, is acidity goes up. So pH goes down when there's breakdown. So that's how we know. And the more it breaks down, the more pink it gets. I should say it's more pink than red. I wrote red because I have red. It's the only, I wish I had more colors in this little app. All right, so the last, and we'll look at lipids in different conditions, like with bile salts. So pancreatin, we're also going to be using bile to like be able to see how that affects things. Carbs. To assess breakdown, we have 2 tests. Lugol's iodine basically shows if there's starch or not. Benedict's shows maltose or other reducing sugars. So we can tell if starch is breaking down because the starch will disappear, there won't be starch left, and maltose, these simpler sugars, will appear. If starch is not breaking down, then the starch will stay and there won't be any of these simpler sugars forming. So we're going to have the ability to both look at the substrates and products of digestion for the carbohydrates. So that's the basic lab. Are there any questions about kind of the basic idea? And we're going to do what we call like an in vitro study, in glass. In vitro, versus in vivo means in a living creature. We're going to basically take test tubes and we're going to throw in our, protein or carbohydrate or fat. We're going to vary the conditions, pH up or down, plus or minus bile if that's appropriate. We'll look at different conditions and see how it affects digestion. So it's going to be a fun way to see how, I can say like, substrate plus enzyme gives you product, but we get to see how that basic process is affected by different conditions. pH changes, presence or absence of bile in terms of fat breakdown. So we're going to do in vitro so we can get a better feel for things that affect the breakdown. All right, what I'm going to do now is show a couple of little videos that just set things up, and then we'll do our breakout rooms. Whoop. Whoop. Wait, this is not what I want to do. Stop. Okay, here we go. Notice my fancy titles.
Okay, we're going to be going over part 1, salivary digestion of carbohydrates. And the enzyme we're using for this part of the digestive lab is salivary amylase. And the source of that is our saliva. So it says to collect 7 mils of your own saliva in a graduated cylinder. So I have close to 8.5, almost 9 mils. And so I've been salivating, collecting it in my little spittoon graduated cylinder. We're going to be using this for not only the spot plate experiment, but also for the 5 tubes of the carbohydrate digestion.
Right, so you guys, last class you missed getting to pee and work with your pee all over the lab, and this time you're getting to miss like spitting and collecting your saliva in little vials and stuff. But yeah, we should also give Becky props for like, you know, a lot of times people even kind of split up, you know, share, like pass it around so you fill it up faster or somebody's, but she did it all herself there. So here again is my lab partner's saliva that we were using as our source of the amylase enzyme for the carbohydrate part of today's lab. Actually, I'll go back to that later. Actually, we'll finish. This is showing just the mucus. So this is showing what happens when you do the, you try to just look at the mucus. We're going to focus on the amylase, but we'll show the mucus here. Saliva has a couple of roles. One is helping break down carbohydrates when you're initially chewing up your food, but it also has mucus that helps kind of bind your food into a bolus to make a nice little slippery ball you can swallow down your gullet. And so we can actually see that other aspect of the saliva. If we take some saliva here, taking a, mil or so of saliva, and this is acetic acid, is basically kind of diluted vinegar, will actually precipitate out the mucins, the mucus part of the saliva. Right, so now use a toothpick. We can see like, ooh, look at that, all those gooey, goopy strings there. Those are the mucins that are the other component of your saliva that are helping kind of bind and lubricate your food. So imagine trying to swallow a bunch of cracker dust. It wouldn't really work. So this is what makes a nice slippery ball go down your esophagus. If you look in detail, totally cool gooey goop in there. All right, so that's nice. So you can see saliva is not just amylase, it's also gooey mucus. So this, let's see, I think this one I start showing how the carbohydrates are set up. We're not going to get to the results. We're going to show the setup here. All right, so now we're going to describe part 1 of the digestion lab. Here, the salivary digestion of carbohydrates. We're going to be using this saliva that was generously provided by my lab mate. And this will be the enzyme. And then we've got our substrate for the enzyme, our starch here. And then we can look at how this process happens under different conditions. So to start, we're going to have to just put some starch. We have 3 mils of starch that we put into each of these 3 tubes. I'm just going to put this up here. Let me go. There's 3 mils in there. 3 mils in there. 3 mils in there. And then also 4 and 5 are going to be controls. Part of an experiment, whenever you're using some kind of an assay, you want to validate your assay. Make sure you give your assay something where you know what the answer is supposed to be, and make sure you get the answer that you're expecting. So then you trust what the answers are when you're looking at your unknowns. So for test tubes 4 and 5, we're going to have test tube 4 which is just going to have pure starch in it. And when we do our test, it better say that, oh, it just has starch in it. Test tube 5 is where we're going to have pure maltose. I might as well just put that in right now. I will put that in a little later. And so when we can check our assay for maltose, that better say pure maltose with no starch. All right, so we have starch in our 3 unknown tubes here. Test tube 1 is going to just fill out our volume by adding 3 mils of water. So 1 mils of water. We got 3 mils of water in test tube 1. Test tube 2, we have to add 3 mils of this quality high grade saliva. And I'm just going to bring this up to 7 so we spit it out. I'm going to spit it out into there. For test tube 3, before we add the saliva in, we are going to increase the pH by using this concentrated hydrochloric acid. This stuff is something you got to be careful with. You should not be using this without wearing eye protection. You know, regular glasses can work. If you don't wear glasses, you got to be wearing goggles of some sort. This can damage your eyes, so, and your skin or your clothes or anything. So be careful with it. We're gonna add 5 drops of the concentrated hydrochloric acid here. 1, 2, 3, 4, 5. And make sure, so now we have this very low pH solution, and now we're going to add 3 mils of our saliva into this. So just note real briefly, it's important to notice that the saliva was not with the starch ahead of time. So it's not like the saliva had a chance to work with the starch and then got acidified. It's like it was really low pH when the amylase was first introduced. So 3 mils of saliva, but the conditions here are going to be very acidic for this salivary amylase to try to do its job. It's important to remember in your mind now that each of these tubes we're going to be testing both for starch using iodine as well as testing for the presence of the products of digestion, maltose, which we're going to use Benedict's reagent for. So we're going to be putting these in the incubator. And after we're done, though, we need to make sure we take each of these tubes and we split it into 2 tubes. That way we can do the glucose test to look for starch in one set, and we can use the Benedict's test on the other side to test for the presence of maltose. If you forget to do this and all of a sudden put your glucose iodine in here without having split it ahead of time, then you're not going to be able to do the test for maltose. So at this point, we're going to take our tubes here and we're going to have to incubate them at body temperature. And we'll come back in a while and see what happens. So here we see test tubes 3, 2, and 1 are joining the test tubes from parts 2. All right, so again, I forget who it was that was mentioning about, enzyme. This enzyme also, it's going to work optimally. It's designed to work at body temperature, which is 37 degrees C. So that's why this bath is 37 degrees C here. So we're in vivo, but we're trying our best to monitor, to mimic conditions in the body. Here, Becky is going to show you what the protein part looks like.
All right, we're going to go over part 2 of the Digestion Lab. This is the gastric digestion of protein. So as you look at the instructions for the lab, the first step is to using a razor blade to cut 5 uniform thin squared slices of cooked egg white. It's important to make sure that these slices are the same size so you can compare the relative digestion. And when we assess the level of protein digestion, we're going to look at how the size has changed along with possibly a couple other factors. So these are uniform. So how these little egg white squares were made was after an egg was hard boiled, the egg white was sliced into very thin sections, put on this graph paper, and then the squares are the same size as the little squares in your graph paper. So if you put a thin slice of egg white, line it up, and then just with the straight razor to go ahead and slice aligned with the squares, and you have the 5 uniform squares. So each of those is going to go into each of one of the 5 test tubes that we are using for this lab. So it'll get in there when we add the various other chemicals. So as you can see from your lab book, we have 5 test tubes that we have labeled 1, 2, 3, 4, and 5. And the egg white is going to go in each of the test tubes along with the different combinations of other chemicals. So test tube 1 is going to have 5 mils of pepsin and 5 mils of water. So each test tube will have 10 mils of different combined solutions. So here is pepsin, and you can see that pipetting 5 mils into test tube 1. And then 5 mils of DI water into test tube 1. So then test tube 2, it'll have 5 mils of 0.5% hydrochloric acid.
No, this is a much weaker acid. This isn't like a crazy. This is just, we're using a weaker acid and base to adjust the pH here, but it's not like the concentrated hydrochloric acid that we had in the first part.
Test tube 2. Along with 5 mils of water. All right, that's test tube 2, test tube 3, and the last tube, test tube 5, will consist of 10 mils of DI water. You should be thinking what test tube this is trying to represent. So we're going to put these tubes in the water, the warm water bath at 37 degrees Celsius. And we're going to put them in for at least a half an hour. And if you can allow more time, an hour is better to assess the protein digestion. So we'll see that later. So these will go in the water bath, set the time. retrieve them in about an hour.
And then finally, here is Tina explaining the beginning of the lipids.
All right, so here I'm demonstrating part 3 of our digestion lab. And the first part is to investigate the properties of bile salts and how they can emulsify water and oil.
And so this first part you should watch. It's kind of like I was showing the salivary mucus in the first part. This is giving you a sense so you can really see what bile salts do in terms of emulsification. And then she's going to set up, but when you do your hypotheses, you think about this emulsification.
And so here I have 2 test tubes labeled A and B. I have added 3 milliliters of water and 3 milliliters of oil. And so the water is at the bottom and the oil is on top. So both test tubes have the same. What I'm going to do now, I'm going to add some bile salts into test tube B. And so you just need a little pinch. And then I'm going to use some of this parafilm, cut some squares. Just going to put some on top here and put some on test tube B. And then I'm going to shake. So we shake for 30 minutes or 30 seconds, and then we're going to just stop. So I'll show you test tube A and B here. And we can compare them right after I've been shaking. So test tube B, this one has bile salts in it. So we're now going to observe what happens over the next couple of minutes and see if we can get the oil and the water to separate out, which we would expect to happen in A, but let's see what happens in test tube B where we added the bile salts.
Okay, so it's almost 1 minute. So 1 minute, let's take a look at them again.
All right, so we waited a minute. You can see the water and the oil they're starting to separate here in test tube A. In test tube B, we have some foam and still an emulsification situation. So we'll wait another minute and take a look at what happens then. All right, so we're going to take a look at our test tubes now after 2 minutes. You can see at the bottom of test tube B, we have a little water down here. It's colored a little brown, yellowish, because there are bile salts also in this water. And you can see that the test tube A, we have the water layer and the oil layer coming back. So we'll wait another minute and take a look at what happens after another minute. All right, so after 3 minutes, we can see that we have water and oil separating in test tube A, and in test tube B, we see an emulsification layer here in the middle, and then water down at the bottom, and then we have some foam up here at the top.
So that emulsification layer, that's the little droplets distributed in the water. As opposed to the test tube A, where it's just basically the oil's back on top and the water's down below. And this is what they put in, kind of salad dressings to emulsify. Like if you want to have like your Bernstein's Italian and shake it up and not have it separate right away, they put emulsifiers in there so you shake it and the oil and vinegar stay mixed up for a while. So. Yeah, so that's, I think, nicely shows what emulsification looks like. And then for the final thing, we'll have her just start to show you how to set up for the lipids. And then we'll go into our breakout rooms.
so next we're gonna do our experiment where we're using Litmus Cream, which is just a heavy whipping cream where we mix some litmus powder in. And so this is a pH indicator. So it's at the start here, it's blue and it turns pink if you add acid. And so I'm gonna illustrate that for you by adding some hydrochloric acid into this test tube, and we'll see how it should turn pink. So, and you can see it's indicating that we've added an acid. All right, so we're going to do the next part of this experiment. So what I have here are 4 test tubes clearly labeled. It's so important that you keep track of which test tube is what getting what treatment. So next, I'm going to add 3 milliliters of Litmus Cream into each test tube. And so I'm going to try to be efficient here and just do it with 1 pipette. Stick it all the way down to try to avoid getting anything on the side of the test tube, and then slowly add 3 milliliters, and then move over to the next and add another 3. And the next. 3, and then I'm going to refill. So it helps to not go all the way to the bottom of this cream because there could be some particles that could clog the nostrils, but let's see. Oops. All right. I'm going to do the last one. And there we go. Great. So next, I'm just going to follow my table and add the various treatments. So for the first one, we have a pancreatin solution here, and I'm going to add 3 mLs of that to test tube 1. Okay, and I'm also going to have 3 mLs to test tube 3. And then the next, test tube 2, I'm going to add a pinch of bile salts and some water, 3 ml. And then test tube 4, just 3 ml water. So the trick here is to not get the test tube wet on the walls here. So when you add the powder, it all just gets stuck on the wall. So I was trying to be careful with that. So next, I'm just going to add my pinch of bile salt to test tube 2. And we're going to try to get it all mixed in too. So next I'm going to add water to test tube 2 and 4. And a pinch of bile salt into test tube 3. So next I'm going to try to get the bile salt dissolved into these test tubes. I have to use some paraffin. Okay, use a stirring rod. Off the walls. Not cross-contaminating my samples. I'm using the clean side of the glass rod in this one. Good. All right, so next I'm going to walk these over to water baths, and it's going to sit there for an hour at 37 degrees C. A little thought question, why 37? What does that have to do with our bodies and physiology? All right, so the temperature is 37 degrees, and then I'm gonna sit here for an hour.
Okie dokie. Blah, 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 blah. All right. So that was a bunch of stuff I see. So are there any questions about the basic idea of what we're doing here? We've kind of shown the setup. I've kind of talked as much as I can and want about kind of the theoretical underpinnings. What I would like people to do now is kind of similar to the kidney lab. I've got all the little breakout rooms ready to go. I would like you in your little breakout groups for each test tube. So for each of, there's 3 different parts, carbs and proteins and lipids. And in each of those, there are different test tubes. So for each test tube, you need to tell me what you expect to find. You know, and in case of the carbohydrates, it's going to be both presence or absence of starch as well as presence of absence of maltose. For the proteins and for the lipids, it's just going to be more or less digestion. And then you need to have, again, the why. A hypothesis always has to have the why. So that was something that people, I think that people did not get as much on when we were doing last time. Always forget, don't forget, maybe I should write it out. Like, you know, the what, oh, hey, goofy thing. The what and the why. I expect to find such and such in test tube 2 because. So make sure you have kind of the reasoning of for each tube, what you expect the results to be and why that makes sense to you. So maybe let's take like 40 minutes to do that. You know, you can also, like in your lab book, when you're writing this down, you know, you saw what happened when you added the acetic acid to the saliva and got the mucus goop. You saw what happened when Tina mixed up oil and water with and without bile salts. So you can like kind of add that into your thing. But so for the next 40 minutes, I want you guys to formally write in your lab book a hypothesis for each tube. And again, work with your lab mates to figure out, you know, to figure out what makes sense, find a consensus. So any comments or questions before I push you all into the rooms? All right, so then I will see you all again at 12:15. Magic. Poof. Oh, we should all go. What did people come up with? Does anybody want to share kind of their predictions for carbohydrates, for instance?
I guess we could share ours for carbohydrates.
That'd be great.
So first test tube, we believe the starch there'd be no evidence of the starch breaking down because there's no enzyme present, no amylase.
Right, so that's right, because starch is just sitting there dissolved in water. There's nothing compelling it to break apart. Starch is actually pretty stable. So that sounds good.
And then second test tube, that we thought the starch will break down and will be shown as maltose due to the presence of amylase in the saliva.
And then did you have a prediction for like the starch test? Never.
I did not. Would you like to include that?
Yeah, since for each of the test tubes, we're going to do both a starch test and a maltose test. So you'd want to think about both of them.
Include starch test.
And what about test tube 3?
So the starch will not break down or only partially due to denaturing of the enzyme from low pH. So I guess both starch and maltose test will test positive, potentially.
Right, so starch, if it doesn't break down, maltose would only be positive if what happens?
The enzyme's present.
Or if the enzyme is functional.
Oh, that's right. Yeah.
Yeah. So if the enzyme isn't working, you might not expect, you probably would not expect maltose to show up. Maltose is only going to show up if the starch breakdown is happening. So good. Awesome possum. What about protein?
Were we supposed to have a prediction for the 4th and 5th control ones? Or what
I mean, you can have a prediction, but I mean, those are, what do they have to be if your test is working properly?
Well, the 4th one, there wouldn't be anything. And the 5th.
What? No, what do you mean? No, of course there's something.
Well, no, like it wouldn't break down because there's no enzyme.
But the idea isn't about breaking down or not breaking down. It's about what results do you expect with those two tests, right? For each of these, you want to, what is the result of the starch test? What is the result of the maltose test? Right, that's how you're evaluating. Those are your dependent variables, is presence or absence of starch, presence or absence of maltose.
So would the 4th one be a positive starch test and a negative maltose test?
Exactly. So that's going to be how we validate our tests. So we know that we actually trust them. And then test tube 5, what about test tube 5?
Negative starch and positive maltose.
Exactly. So it's always an important thing when you have an unknown that you're using a test for, that you check out your test on something where you know what the answer is supposed to be and make sure that you get that answer. I think I might have shared, like, one of the hilarious classic examples of people not validating their test was some of the early stuff that was saying LSD breaks your chromosomes. And they had like some little test where they put LSD into this test and showed evidence of chromosome damage. And then they never validated the test though. So that once people looked at their technique for doing this analysis, they realized, oh, milk breaks the chromosomes, tap water breaks chromosomes. Like the test wasn't actually telling you anything interesting. So you need to make sure that you use the test on something where you know what the answer is supposed to be and make sure the answer is what you expect. So then you can trust the results more when they're doing the unknowns where you're not actually sure what's really going on. So yeah, so cool. So that's a good question. So those are important there just to validate our, and sometimes they're not correct because there's been some contaminant in the starch solution or something or other. It's not always the case that those come out the way you expect. And when they don't, that means like you need to figure out what happened. And it also means you can't trust your other results. So, all right, what about protein? Anybody? We had Stephen volunteering for carbohydrates. Maybe,
Okay.
okay.
So for test tube 1, we said that the size will remain the same because the protein will not break down. It will not break down the protein because the neutral pH of water will denature the enzyme pepsin.
Okay, so that's good. Remember, because pepsin is one of these enzymes that's actually optimized to work in a very acidic pH. So for it, being neutral is actually unpleasant. So it's definitely not its optimal pH. Let's continue on. What about test tube 2?
So for number 2, we also said that it will remain the same size because there's no enzyme to break down the protein.
So good. So without an enzyme, so perfect. And
then number 3 was more exciting. We thought that it will get smaller because the pepsin will be able to function in the acidic environment of the hydrochloric acid.
so it's good. So you're on a roll. Why don't you just go all the way to number 4?
Okay. Number 4 will remain the same because the base from sodium hydroxide will denature the enzyme pepsin, which needs an acidic environment to function.
All right. So, and then yeah, the 5th one was just kind of in water, so you could compare to the other ones. So perfect. So that's, that's really nice for a hypothesis, kind of short and sweet, it's to the point, but it's like very clearly what do you expect and very clearly why that prediction makes sense. So, and finally, lipids. Who's going to go for lipids? I'll do is I'm gonna like put my finger down on the thing if nobody answers. All right. I have Megan B. Oak. Is Megan Oak still here even? We've lost Megan Oak. Gerson E. Garcia.
Yes, for tube number 1, we said the Litmus will turn pink. And it will just wait. What is it? Wait. There will be like just wait, just let me. We wouldn't have as much of a breakdown because there's not a bile salt, so it will be harder to break down with a, yeah. Does that make sense?
Yeah, so that was test tube 1.
Yeah.
Yeah, so you're gonna have breakdown because you've got the enzyme, but without bile, it's not going to be as efficient. Although because I mentioned cream actually is an emulsion to start with, it's not like it's not going to be able to work. You're still going to obviously get breakdown. So what about test tube 2?
There will be no breakdown because there's not an enzyme.
Exactly. So that's important to remember. Bile helps by emulsifying, but it's not going to actually create an actual molecular breakdown. So there's not going to be any acids coming into being. And what about test tube 3?
Yeah, we'll see a breakdown because the presence of the bile salt will make it easier.
Yeah, how would you compare that to like 1, where you also expect to break down?
Because of the bile salt makes it easier for the pancreatic to function.
So you're probably gonna, you'd expect there's gonna probably be breakdown like there was, but it's probably gonna be even more intense, I'd expect.
Yeah.
So, and then what about test tube 4?
No breakdown because there's not an enzyme.
Okay, good. All right, so is everybody, even those the ones of you that were shy, hopefully that all kind of makes sense to you. In terms of the results, we actually, the videos have us doing the stuff, but I can actually, in the interest of time, given that it's 12:26, I'm going to kind of cut to the punch here. And doing the lab. Okay, so if you're interested in watching me boil the Benedicts, or Tina go and look at the stuff more intensely, I recommend you do watch these. But for right now, in the interest of getting you out in time, and because I actually have a department meeting, I'm going to publish. So I kept these results hidden from you so you wouldn't see them when you were doing your what do you call it? Doing your hypotheses. So Digestion Lab Results are now there available for you. And basically, part 1 for Lugol's test and Benedict's test. For each tube, we split it into 2 and took a look at what happened in both starch and in maltose. And we see test tube 1, the one that was starch with just water. What do we see in terms of Lugol's? Can you see this?
Is
What
that
is it?
dark purple?
Yeah, what does Lugol's mean if it's dark purple?
Presence of starch.
Yeah, Lugol's is that dark purple black if starch is present, and it's more of this kind of amber if there is no starch. So whenever you see it being black, it means starch. Whenever it's amberish, no starch. So this is saying lots of starch present. If we go to Benedict's, Benedict's is blue if there is no sugar, and it turns other colors, green to orange to red, if there is sugar. So this is saying test tube 1, lots of starch. What about maltose?
It's negative.
It's negative. Benedict's, if it's blue, is negative. So no maltose, but yes, starch. Does that make sense? Yeah, because we didn't have anything to break down the starch. Look at test tube 2 here. How much starch is left after you've had it a half hour with your spit? Can you see is it black or is it amber?
Or amber.
Or amber. So that means there's no starch. Again, Lugol's is going to be that purple black if there's starch. It stays kind of just amber if there's no starch. So there's no starch. Where did the starch go?
broke apart.
into,
Maltose.
yeah, and take a look. Look what we have here down at the Benedict's for that tube. All of a sudden we see the presence of the maltose. This is very direct evidence that the starch was converted to maltose by the saliva. What about test tube 3? That was the one that had the exact same thing, saliva and maltose, except under the really high pH. What do we see here?
The presence of starch because the enzyme couldn't break it down.
Exactly. So starch is still there. It didn't dissolve, it didn't break down, and there's no starch showing up at all in the tube. So this is almost as if the enzyme was never there, right? And then our controls are validating our test, right? The starch is saying positive for starch, negative for maltose. The maltose is saying negative for starch, positive for maltose. So that all looks good. That makes sense? You know, for your exam, you should be able to understand the color as well. Like on an exam, I would feel perfectly fine showing you tubes, and you have to understand what's going on. Like if I say this is a Lugol's test or the Benedict's test, you need to know what that means, if it's black or if it's amber, if it's blue or if it's orange. So make sure that you understand the test, how the test works.
Maltose is a disaccharide of 2-glucose.
Yes, exactly.
Is that right? Okay.
Exactly. Yeah, the amylases tend to, if you remember, starch was a bunch of glucoses all in a big chain. Amylases don't cut it apart into individual sugars. It kind of cuts off the big carbohydrates into smaller chains. And there's going to be other enzymes later on that do the final fine tooth comb breakdown into individual monosaccharides. All right, so I'm going to clear this. Let's take a look here at the lipids. And what we see here, did breakdown occur in test tube 1?
Yes.
Yes, we see it's turning pink. And yet in 2, we can see it stayed blue. This is the one with just bile salts. So bile salts alone, it just stayed blue. This one was just cream and water. Obviously, nothing broke down. What about tube 3?
It looks more acidic.
Yeah, it looks even a little brighter pinky than 1. This is the one where we added the bile salts. So it looks like the bile salts helped actually even increase the breakdown. So this is pretty nice. This result is pretty nice. So does this make sense? All right, so the final thing I'm going to show you guys here. Oh, I want to clear, hold on. I got to finish up here. This file here, this is actually data from last semester. Oh, I need to shoot. I need to share it. There it goes. So this is just showing the carbohydrates, 1, 2, 0, meaning it would be not, it would be negative, 1, 2, 3, the number of pluses. So this is just kind of showing across different groups what happened in the different tubes. So when you compare, when you talk about our individual results, you can also compare to the overall results to make sure everything's consistent. Same for lipids. For proteins, these were the results that they found. So you can use, I could not get good pictures of the protein tubes. It was really tricky to get anything that looked useful. So you can just use these numbers here to say what the results were from the protein. And it's a little ambiguous. It's not as nice as you would hope it to be. So, and that's part of science. So that's one of the things that's really frustrating about doing these dry labs versus about doing real labs is you don't get as much of a sense of how messy and tricky it is to do good science. So are there any questions about this lab before we bid thee all adieu?
Can I just ask about the quiz on Wednesday?
Yeah.
What, just up till now, what we study?
Yeah, everything through not the digestive system, but definitely the immune system. Yeah, so good question. Yeah, so we'll do the quiz on Wednesday, and it'll just be kind of 10 points, 10 like multiple choice answers looking at the immune system and oh my God, what else were we doing?
Kidneys.
The kidneys. Yeah, so urinary system and the immune system. So otherwise, I hope you all have, it actually looks like a beautiful day. So minimally you can like have sun streaming in your window or maybe even walk outside. And I will see you all. And I have office hours, but not till 2 because I actually go to a department meeting right now. So I will see you all then.
Thank
Thank
you.
you.
Thank you.
Thank you.
Okay,
Bye-bye.
bye.
Let's see. So mast cells are part of the triggering of inflammation.